Hi, welcome back. In this video, I'm going to look more closely at the two main phases of photosynthesis. As you can remember from the last video, photosynthesis can be broken down into two main phases or two separate reactions. The first being the light-dependent reaction, and the second being the light-independent reaction. If we take a look at a diagram, we've got our little plant cell here, and you can see the various chloroplasts found within the plant cell. And more than likely, if we're talking about the cells of a leaf, these would be the either spongy or palisade mesophyll cells. So if we were to take a closer look at the chloroplast, we can see the outer and inner membrane. We can see a granum right here with lots of thylakoids. From the thylakoids are the little green discs. And then this orange fluid on the inside will be the stroma. So the light-dependent reactions take place along the thylakoid membrane or in the thylakoids. You can see that the thylakoid right here is capturing solar energy using chlorophyll or one of the accessory pigments. Water is used in the light reaction, and it is split, and oxygen is released as a waste product. Light reactions is also going to produce NADPH and ATP. NADPH is going to provide electrons for the Calvin cycle, which is another name for the dark reactions or another name for the light-independent reaction, right? It's kind of a series of reactions. It's got lots and lots of names, but I like light-independent. And ATP is going to provide the energy, right? And the light-dependent reaction is going to use the energy in the electrons from the light-dependent reactions to fix carbon dioxide into a carbohydrate. And then the NADP plus and the adenosine diphosphate will return to the light reaction to be charged again. So it is very cyclical, right? In fact, this is cyclical photosynthesis. Now, let's look at the light-dependent reaction, or the LDR, right? The light-dependent reaction. This is going to occur, and I'm going to go ahead and use green. This is going to occur along the thylakoid membrane, right? And now remember, the thylakoid membrane, right, is going to be the double layer of phospholipids, right, that make up those little green discs, right? So this would be the inside of the thylakoid. This would be the outside of the thylakoid. And if you remember from our diagram, the outside surrounding the thylakoids, this area is going to be the stroma, right? It's that liquid solution where the light independent reaction takes place. And in some textbooks, you'll actually see this inside area is referred to as the thylakoid lumen. You'll see this term used a lot in biology, just talking about the inner portion of something. Usually the inside of an organelle will be called a lumen. So what's going on in the light-dependent reaction? Well, the light-dependent reaction involves the capture of solar radiation or photons. And this is actually accomplished by photosystems, as I had outlined in another video. These photosystems contain chlorophyll or other accessory pigments. And believe it or not, there are actually two of them. This one right here, the one we'll start with, is actually the photosystem number 2, or PS2. And then the last one is PS1. You might be asking yourself, what in the world? Why are they out of order? Well, the photosystem number 1 was discovered first, and then photosystem number 2 was discovered second. So the photosystem 2 is responsible for absorbing a photon, right? So it's absorbing solar radiation or a packet of light. And the photon excites chlorophyll. It excites an electron found in this photosystem. And this little electron, remember electrons are negatively charged, he gets so excited and he bounces out to an excited state and can easily bounce off of the molecule, right? Because of this addition of energy from light. And this excited electron can actually zip across this long chain of proteins. There's lots of them, right? I'm just going to draw them as one big chain, right? And it's called the electron transport chain, right? ETC, electron transport chain. What does it do? It transports an electron. So this excited electron gets boom, knocked into by sunlight. He gets really excited and he zips across this electron transport membrane. And then when the electron gets over here to photosystem 1, photosystem 1 has chlorophyll and other accessory pigments as well, and it's going to absorb another photon of light. It's going to absorb some more sunlight, and it's going to re-excite this electron, right? So this electron was kind of losing some steam. It gets zapped by another photon, and he gets excited again, right? Rises to a more energetic state, and then he gets caught by a really interesting enzyme known as NADP plus reductase. And I need to find a better way of doing this. This is really hard to write with a mouse. I don't know if you guys ever had to do this. NADP plus reductase. How do I know it's an enzyme? Look at that suffix, ASE. So this is an enzyme, and what does it do? It reduces NADP plus. Well, if you've got a molecule that's got a positive charge and it captures or grabs a bunch of electrons, what do you think is going to happen to that charge? It's going to be reduced, right? Now, what does that mean? Well, it means that we can take NADP plus, and this molecule has a really crazy long name that we're not going to bother with, and the NADP plus reductase drops a bunch of those electrons off and allows NADP plus to become NADPH. And so now this molecule has the ability to carry a bunch of extra electrons as well as a hydrogen molecule. And sometimes in science, if you see hydrogen all by itself, right, it's written like this, and we call that a proton, right? And so there's a proton floating around here in the stroma that gets snagged up by NADP plus once it's reduced by NADP plus reductase, right? Now, if you remember, what is another product of the light-dependent reactions, right? So far, we've produced NADP plus. Well, there's two other things we need to make. We need to make oxygen, and we need to make ATP. Well, let's first start with oxygen. Where's the oxygen coming from? Well, if you go all the way back here to photosystem 2, the sunlight excited that electron. The electron ripped off the photosystem and zipped across this little electron transport chain and then got bounced up again and then eventually ended up over here. So now this photosystem 2 is missing an electron. And if we want to make more, we need to replace that electron. So the photosystem 2 performs what is known as photolysis. That's a pretty incredible reaction. Photolysis, photo meaning light, lysis meaning to cut or to break. Photolysis takes a molecule of water and breaks it apart. It breaks it into some hydrogen ions, also referred to as protons, and oxygen, right? And it does this by ripping an electron off of water, right? So it rips electrons off of water to kind of replace that electron it had lost to this crazy little journey. And we're going to produce some hydrogen, protons, and oxygen. And the chloroplast doesn't want the oxygen, doesn't need it, and so the oxygen can be released as a waste product. Now, as you can imagine, over time, we're breaking down lots of water molecules to replace lots of electrons, and the oxygen gets released, but these H pluses, these hydrogen protons, they start to accumulate in here, right? And we end up with a bunch of them in here, and this will become important here in just a second, okay? Now, so there's our oxygen. There's our NADP plus, or sorry, NADPH, and we're missing one other product. We're missing ATP. So how in the world are we going to make ATP? Well, there is another enzyme embedded in the thylakoid membrane, and it is known as ATP synthase. I wonder what it synthesizes. It synthesizes ATP, hence the name ATP synthase. ASE lets me know it's an enzyme, and then synth for synthesis. And what are we synthesizing? We're synthesizing ATP. So how does this enzyme work? Well, this enzyme takes molecules of ADP and it forms ATP. Now, if you remember, if we think about ATP as our battery, right, this would be a battery with 3 bars, triphosphate, and this is 2 bars, diphosphate. Well, we need to add another phosphate here, right? And this is going to require energy because if you remember from a previous video, I talk about the energy. It's being contained in the bonds between the phosphates. They don't really want to be close together, so they have to be squished together. So where is that energy going to come from? Well, ATP synthase actually relies on something known as a chemiosmotic gradient, right? Which simply means you got a lot of something or a lot of chemicals in one area and they want to naturally flow from high concentration to low concentration. And ATP synthase takes advantage of this growing number of hydrogen protons in the thylakoid lumen. They just keep accumulating and keep accumulating and keep accumulating. And they want to get out because they're all positively charged. They don't want to be next to each other. And ATP synthase provides them a path, an opening, right? A specifically shaped protein opening that allows these hydrogen protons to escape, right? And what's really neat is the thylakoid uses the electron transport chain to add even more hydrogen protons to the lumen, right? So up here, when these electrons are excited and they move across this electron transport chain, it's almost kind of like electrical current, and it powers this little protein right here, this little proton pump, and it takes hydrogens from out here in the stroma, these protons, and it pumps them into the lumen. So we have two sources of hydrogen ions, two sources of proteins. So you could imagine, right, as this continues to cycle over and over and over again, these hydrogen protons start to get really packed tightly in the lumen. They want to escape. And as they zip out of this ATP synthase enzyme, they provide the energy to take adenosine diphosphate and to form ATP. Okay, and so that's the light-dependent reaction. I know that's a lot. Do you think I'm going to have you memorize all these different photosystems, these different enzymes? No, but you should be, on an elementary level, able to understand just simply by recognizing the name. What does ATP synthase do? It's an enzyme that synthesizes ATP. What does NADP plus reductase do? Well, ACE, it's an enzyme. It reduces NADP plus, right? You do need to know, where does the oxygen, right, released by photosynthesis, where does it come from? It comes from photolysis, which is the oxidation of water, right, the breakdown of a water molecule. And let's make sure we've got everything that we need. Okay, I see here's our product of the light-dependent reaction. We've got our oxygen, we've got our NADPH, and we've got our ATP molecule, okay? Awesome. So now we have everything we need to start the light-independent reaction, the LIR, the light-independent reaction. The light-independent reaction is going to take place in the stroma, so it's going to take place outside of the thylakoid, okay? And the light-independent reaction relies heavily on a lot of enzymes, but one enzyme in particular that I absolutely adore its name, and that is Ribisco. I think if I ever got another dog, I would name it Ribisco. And this is an abbreviation for a long name. It's ribulose bisphosphate coenzyme, right? So it's an enzyme that actually participates in fixing carbon dioxide, right? And so how this works is we've got carbon dioxide, right, the gas in the atmosphere that is going to be absorbed through those tiny openings in the bottom of leaves known as stomata. It'll enter the leaf, eventually enter the mesophyll cells, find its way to the chloroplast, where it'll float around in the stroma. And then the carbon dioxide will be fixed by Ribisco, right? And Ribisco takes a 5-carbon sugar, and I'm just going to write 5-carbon, right? It's got a different name. It's ribulose bisphosphate, right? It's a 5-carbon sugar. And Ribisco takes this 5-carbon sugar and it attaches a carbon dioxide molecule, right? And so then we end up with, a 6-carbon molecule. And then this is where it gets kind of complicated, right? So this first part right here is carbon fixation, right? We've taken carbon dioxide and we've fixed it into a molecule, right? And the next part is the reduction phase. And the chemistry here is kind of complicated, and luckily you don't have to worry about it. But this is where both ATP and NADPH get involved, right? They get involved in this part. And they help energize these carbon molecules, make them just right for producing glucose, right? And so we end up with, right, with 6 carbon sugar. It actually gets kind of split and energized here. We end up with 2 3 carbon molecules, right, that can be easily combined to form our carbs. And believe it or not, right, these 3 carbon compounds right here can actually be used to form a lot of things. But right now, we're just going to think, yeah, these are going to be used to form glucose, right? And that makes sense because if you take 2 3 carbon sugars and you add them together, you're going to get glucose, right? C6H12O6. Now, not all of these little 3 carbon chunks will be used to make carbs. Some of them get recycled back into that 5 carbon sugar that is needed for this whole reaction to take place. So you have a carbon fixation, right, where carbon dioxide's fixed into that 6 carbon sugar, and then ATP and NADPH, take our 6 carbon sugar and split it in half and get it just right for forming glucose. And then some of those extra 3 carbon sugars are going to be used to reform that 5 carbon sugar that Rubisco used initially to fix carbon dioxide. And I know that's really complicated, and this is a very simplified version of the actual reaction, because actually over here for 3, to get this 5 carbon sugar back into working order, we actually would need to add some more energy. We need to pack in some more ATP, right? And then they're thinking, oh my goodness, this is all giving me a headache. Really? But at the end of the day, what does the light independent reaction produce? It produces carbs, right? And so it's just a cyclical enzymatic reaction. So lots of enzymes are involved right here, right? Taking carbon dioxide and capturing it out of the air and fixing it into a usable form with which with which plant cells can use to form carbohydrates. Well, that's the light-dependent reactions and the light-independent reactions in a nutshell, right? And hopefully this provided a basis for understanding the mechanics. Are you going to have to understand all of this memorized? No. You need to really make sure that you understand, let me go back a slide, where they take place and what they're using. Light-dependent reactions are in the thylakoid. They produce oxygen, NADPH, ATP, which the NADPH and ATP is used by the light-independent reactions to fix carbon dioxide into carbohydrates. And you can see why it's a cycle, right? Because as the NADPH drops off some electrons, right, it returns as a positively charged compound to the light-dependent reactions. ATP drops off some energy, right, by breaking some of those phosphate bonds, and returns to the light-dependent reactions as adenosine diphosphate. Well, that's all I have for this video. The next video will look at the various environmental conditions and factors that either increase or decrease the rate of photosynthetic reactions.